Hello everyone. Today's talk will be based on this recent article we published in the Lancet Family Open Access Journal E-Clinical Medicine titled Evaluating the Evidence Behind the Surrogate Measures Included in the FDA's Table of Surrogate Endpoints as Supporting Approval of Cancer Drugs. This is an open access article, and the link to download this article is provided at the bottom of the slide. Most of the data that we discuss on today's article and the discussion related to that you can find in the full text of this article. Thank you.
So good afternoon and welcome to the National Breast Cancer Coalition's LeadCast webinar with our featured presenter, Dr. Bishal Gayawali. My name is Michelle Tregear and I'm the Chief Programs Officer at MBCC, and we're pleased that you could join us today, and we look forward to an engaging presentation followed by active discussion. Before we begin, let me just say a few words about the webinar and provide some logistical details. First, today's webinar is being recorded. Both the presentation and following discussion will be archived, and a link will be available after the presentation. You've also been automatically muted so that we can minimize background noise. Dr. Gayawali will present for the first portion of the webinar, and then we'll use the remaining time for questions, answers, and discussion. Now, if you'd like to ask a question when we get to that point, you can raise your hand by clicking the hand icon at the bottom of your screen, and we'll unmute you. And you can also ask your question throughout the presentation using the Q&A feature at the bottom of your screen, and we'll try to answer as many of those questions as we can. Now, I'd like to introduce today our moderator, who is Judy Hirschfeld-Bartek. So Judy began her career as a clinical nurse specialist in all areas of breast oncology until her retirement in 2018. Judy's personal connection with breast cancer includes her mother, grandmother, and cousin, who are all diagnosed with the disease. She has served on the board of the National Breast Cancer Coalition since 1992 and presently represents the Dr. Susan Love Research Foundation. She has been on several MBCC task forces, including the clinical trials, clinical care, and genetic testing. And Judy is a graduate of Quality Care Project Lead and is a past president of the Boston chapter of the Oncology Nursing Society. She is committed to the Artemis Project and believes that advocates must be part of any initiative to end this disease. So at this point, I would like to turn it over to Judy.
Thank you so much, Michelle. I'm so excited about this, this LeadCast, especially after we had that fabulous summit and the session on the perspectives on oncology drug approval, which touched a little bit upon the idea of surrogate markers as endpoints and the impact in breast oncology trials. And this is going to be even more information on that, and I think it's going to be very exciting. So I'm really, really happy. And I'm pleased to be able to introduce you to our esteemed presenter for today's webinar, Dr. Bishal Gayawali. He is a practicing medical oncologist and scientist in the Division of Cancer Care and Epidemiology at Queen's University Cancer Research Institute in Kingston, Ontario, Canada, where he is also an assistant professor of public health sciences. Prior to his position at Queen's University, he served as a fellow in the program on regulation, therapeutics, and law, also known as Portal, at the Harvard University. And you might remember, if you were on the plenary session on the drug approval process, Dr. Darrell also spoke from the Portal program at Harvard University. His clinical and research interests include cancer policy, global oncology, evidence-based oncology, financial toxicities of cancer treatment, clinical trials, and supportive care. Dr. Gayawali is an advocate of the cancer round shot, a term he coined to imply that research investment must be made on known high-value interventions in cancer care that are affordable and easy to implement globally. Dr. Gayawali is also and active in the oncology and clinical research communities on Twitter. And if you are on Twitter, I highly recommend that you follow him on @oncology_BG. Today, Dr. Gawali is going to be talking about his work regarding FDA surrogate endpoints in breast cancer, and we are really, really pleased and honored to welcome Dr. Gawali to this website. And now I turn it over to him.
Thank you very much for the kind introduction and invitation. It is my huge pleasure to be back again with the NBCC LeadCast program. Some of you may have heard me before. I had presented a webinar before in January 2020, and now I'm really excited to be back and to be discussing a little further about the concept of surrogate endpoints today. And as I have always said, I think, and I echo what Judy just said, I think patient empowerment through education is quite important in order to change any cancer policy. And that's what has been my personal experience as well. No matter how much we speak with regulators, policymakers, people who can make changes, it had some impact. We write papers, we give presentations at ASCO, ESMO. It has some impact, but the biggest impact has always come from speaking with patients and changing patients' perspectives are helping them to understand these things. So it is always my big pleasure and honor to be speaking with patients and patient advocates. So thank you very much for this opportunity again. So today, I'd like to talk about the FDA table of surrogate endpoints and its use in regulatory approval. First of all, I'd like to disclose that I have no conflicts to disclose. And before going into the FDA table of surrogate endpoints per se, which is the main focus today, I'd like to give a brief background about the idea about surrogate endpoints and regulatory approval by presenting some of the slides that I had presented earlier in January. So first, the fundamental question is that all drugs, especially cancer drugs, they have many side effects. And some of these side effects can be quite serious or even fatal. So the question is, then why, and of course, these are all super expensive, then why would a patient with cancer be willing to take those drugs? The answer is simple, because they want to either live longer or live better, ideally both. They want to live longer and better. If not both, at least one, have a better life or a longer life. In terms of the language of trial, we call this either the drug has to improve survival or the drug has to improve quality of life. Ideally, the drug has to improve both. If not both, either one of them. So that's quite simple. That's the reason why a patient is willing to take all those drugs. But in cancer trials, we see increasingly that we are not testing these metrics. We are testing something else. We are not testing whether a patient lives longer with these drugs or not, or a patient lives better with these drugs or not. We are testing something different. And that something different, that proxy, is called a surrogate endpoint. So, for example, some of the commonly used surrogate measures in oncology include response rate. So a response rate, for example, what it means is we give a drug to, let's say, 100 patients, and out of those 100 patients, we see how many patients will have a tumor shrinkage. And if 30 patients out of 100 will have a tumor shrinkage, then we say the response rate is 30%. And how do we define tumor shrinkage? It's quite arbitrary. We define it as at least the tumor has to shrink by at least 30% or more. Now, why we call it a surrogate is we do not know whether shrinking tumor is going to lead to a longer life or a better life or not. It may not. And that 30% is quite arbitrary. There is no logic as to why a shrinkage of tumor by 29% would not be a response, but a shrinkage of tumor by 32% would be a response. And the same is true for progression-free survival. Progression-free survival, although this term has the word survival at the end, it does not exactly measure survival only. It measures it is actually duration of progression-free interval. And by progression-free, we mean the duration until which the tumor does not progress. And by progress, again, we mean an arbitrary cutoff where the tumor increases in size by more than 20%. Again, an increase in tumor size by 19% is not progression, but an increase in tumor size by 22% is progression because of that cutoff limit of 20%. And these are some of the reasons why improving these surrogate measures may not translate into improving clinical benefit for the patients. But the question is, why do we use these surrogate measures then in trials? And of course, there is a reason why we want to use this, because measuring survival takes quite a long time, or measuring quality of life is complicated. So surrogate measures provide a very attractive option, because it takes, let's say if the survival time on average is 3 years, then you do not have to wait for 3 years for the drug to get approved. And of course, the patients need access to good drugs as soon as possible. But if you use response rate, then you can see whether the tumor has shrunk or not in a relatively very short time. So that is the reason why using surrogate endpoints look very attractive for clinical trials. But this is the hypothesis. But the question is, how much time do we actually save, or are we actually saving in oncology by using surrogate endpoints rather than overall survival? And this paper actually looked into that question, and the result they found was, after looking at 107 oncology drugs, they found that using progression-free survival instead of overall survival would save 11 months and for the total trial completion and approval. And using response rate instead of overall survival saved 19 months. Now these are not nothing. Like these are important time saving. Like 11 months time saving by using PFS, and 19 months of time saving by using response rate. So that means a patient will have access to a drug that is approved on the basis of PFS 11 months earlier compared to if we had waited for overall survival. But sometimes in the oncology community, if like people seem to make it as if it's a difference of 5 years or 3 years. People say, oh, we can't be waiting for 4 or 5 years for overall survival. If we use response rate, then we'll get answer quickly. But what data actually show is that it's not that dramatic of a difference. It is a difference. It is important, a saving of 11 months or 19 months, but it's not 4, 5, 10 years as people make out to be. So we have to keep this equation in mind when we are considering the trade-offs of surrogate endpoints, a time saving of 11 months with PFS. But the other question is, why are we not using surrogate endpoints all the time then? Why do we actually want to use overall survival? Because and the answer is, as I hinted earlier, those improvement in surrogates may or may not translate to improvement in clinical benefit. For example, this study looked at correlation between improving a surrogate endpoint and improving overall survival. And they found that of the 89 settings in which this was studied, only in 12% was the correlation high. That means improving surrogate also predicted improvement in overall survival. But in most other settings, these blue bars represent low correlation. So in most of the settings, improving a surrogate endpoint did not translate to improvement in overall survival. It was not predictive of improvement in overall survival. And that's the reason why we need to exercise caution in using a surrogate endpoint, because that may not predict clinical benefit. The other question is, what about quality of life then? Is overall survival the only endpoint? Of course, we discussed at the outset that overall survival is not the only clinically meaningful endpoint. Quality of life is equally a clinically meaningful endpoint. So the natural hypothesis is, if we improve progression-free survival, then that should improve quality of life, right? because the patient is without disease progression for a longer time. That's quite a natural hypothesis. But as all hypotheses, this has to be tested. And we did try to test it in 2018. And what we found was we looked at 352 phase 3 randomized controlled trials, and we found that 50% of these trials, they did not even include quality of life as an endpoint. Nobody bothered to even study quality of life as an endpoint. And of those that did include quality of life as an endpoint, 25% did not report quality of life. So this means these are trials that included quality of life as an endpoint in the design, collected quality of life data from patients, but decided not to report it. Why does that happen? I'm assuming because the quality of life data were not good. Otherwise, they would have presented. So altogether, if you look at, and these are only 352 phase 3 RCTs in one calendar year that we studied. So altogether, there were more than 125,000 patients enrolled in these studies, whose quality of life outcomes we do not know. And this is a huge waste or misuse of resources. More than 125,000 patients. We could have known quality of life outcomes for so many drugs, but we do not. That's one part of the story, but the important part about surrogacy that I want to talk here is that when we looked at whether improving PFS led to improvement in quality of life, we found that the correlation was quite weak. So improving PFS did not necessarily translate to improving quality of life. So from the previous slide, we saw that improving PFS or other surrogate did not necessarily translate to improving survival. And from this study, we know that improving PFS does not necessarily lead to improving quality of life. And that's the reason why we always have this debate in cancer trials that use surrogate endpoints. So what about the role of surrogates in regulatory approval? Of course, there are always two aspects to all the questions. And I personally think that surrogates do have a role in regulatory approval. It's not like they are nothing. They do have an important role, but we have to consider always the two aspects, the false positive, the risk of false positive and the risk of false negative. If we look at the patient population, we also find two groups of patients. A group of patients wants access to cancer drug as soon as possible. They do not necessarily want to know whether it is really effective or not. They are ready to accept a higher level of uncertainty in benefit. But then there is another group of cancer patients who sometimes they say that I don't want any cancer treatment at all. I'm afraid of chemotherapy, or like even for effective treatment that we know overall survival, they are quite scared to take any cancer treatment. So there is one group of patients who say, I want to take cancer treatment if you are absolutely certain that receiving this drug is better for me. It will help me live a longer life or a better life. So we have to, as a regulatory agency, I think the regulator have to balance both these two group of patients' expectations and concerns. So I think in that regard, the accelerated approval system in the United States, the FDA, is a very good compromise between the need for earlier access and the need for a confirmation of benefit. So what accelerated approval gives us is it will give earlier approval on the basis of surrogate endpoint. So the drug can be approved. So the patients who want earlier access can have access to the drug, but it will also mandate a confirmatory trial to be conducted post-approval. And that confirmatory trial should confirm clinical benefit on a clinical endpoint. And if it fails to do so, then that drug can be revoked. The approval can be revoked. So that's a nice compromise. But the whole idea of a couple of concerns regarding accelerated approval is that the whole idea of accelerated approval is to give earlier access to cancer drugs, but the pricing mechanism is such that the price of drugs that are approved on the basis of improvement in overall survival and the price of cancer drugs that are approved using this accelerated pathway on the basis of surrogate are the same. Or sometimes even these drugs cost higher than the drugs that have demonstrated improvement in survival. So those high cancer drug prices, now it will limit access to the cancer drugs. So the whole idea of accelerated approval is to increase earlier access to cancer drugs, but because of the pricing, the access to cancer drugs is already compromised. So I think, and in this paper, we argue that at least the pricing of cancer drugs should match the level of evidence, and accelerated approval cancer drugs, because they may or may not improve clinical benefit, should have lower drug prices. But that's a different part of the story. Today, let's focus on regulatory approval and surrogate endpoints. The other problem with accelerated approval, I was trying to mention, is that a lot of these confirmatory trials, they do not confirm clinical benefit. So what happens is that a drug gets accelerated approval on the basis of surrogate endpoint. A drug is mandated to be tested in a confirmatory trial, but A, the confirmatory trial does not happen. B, the confirmatory trial happens after a long time, after 7, 8, 10 years, and the drug with uncertain clinical benefit remains in the market for such a long time. And C, even when a confirmatory trial is conducted and reported, the confirmatory trial actually does not test for clinical benefit. This is what we found in our paper. Of the 93 cancer drugs that had received accelerated approval, the confirmatory trial status was ongoing for 10%. It was pending for 11%. It was delayed for 5%. And the confirmation of benefit happened only in 62%. And now 62% looks like a big percent. It is. But if you look at this confirmation of clinical benefit, the benefit in clinical outcome in overall survival was in only 20%. The rest of the clinical benefit, confirmation of benefit, was in a surrogate outcome. And the surprising thing was, for some drugs, the confirmatory trial tested a surrogate outcome, the same surrogate outcome that was tested in a pre-approval trial. Now, this defies logic to some extent, because if it is the same surrogate outcome, then at the beginning, you thought that this is a surrogate outcome that can only lead to accelerated approval, not full approval. So you gave accelerated approval. But then again, in a confirmatory trial, you test the same drug in this using the same surrogate outcome, and now you are saying that, okay, this confirms clinical benefit, and it confirms the mandate for a confirmatory trial. So this is where the problem is. And the other part of the problem is, you can see that there are 8 drugs that did not confirm benefit. They actually failed to show clinical benefit in a confirmatory trial. But some of them, they stay on the market and are not necessarily withdrawn. The idea of accelerated approval is to withdraw the drug if it does not confirm clinical benefit. But what is happening in practice is it fails to confirm clinical benefit, but it still stays on the market, like in the case of bevacizumab in glioblastoma. Bevacizumab in breast cancer is a good example of how accelerated approval should work. Got approved earlier on the basis of PFS, in a confirmatory trial, failed to improve OS, got revoked. That's how it should function. But you look at bevacizumab, the same drug, but in glioblastoma, it got approval on the basis of response rate in a phase 2, was tested in a phase 3 using overall survival. And as you can clearly see here, overall survival did not improve. Quality of life was also tested in this trial, and quality of life was also not improved. But still, it got full approval. So that is where the problem is. Now, this is from the FDA's own website. I have a link here at the end of the slide if someone wants to see this. So what does the FDA itself says on its website is that clinical trials are needed to show that surrogate endpoints can be relied upon to predict or correlate with clinical benefit. This is the FDA's own mandate. Surrogate endpoints that have undergone this testing are called validated surrogate endpoints, and these are accepted by the FDA as evidence of benefit. So this is what the FDA says. Clinical trials should show that surrogate endpoint is actually a reliable surrogate. It correlates with clinical benefit. And those surrogates that are tested in this way are called validated surrogate endpoints. And so please keep this language from the FDA itself in mind as we move forward. So a recent development that I want to talk about today actually is that the FDA has published a table of surrogate endpoints. that were the basis of drug approval or licensure. So in the past, as well as the surrogate endpoints, they can use in the future. So FDA, this was one of the mandates of the 21st Century Cures Act. So the FDA has now on its website published a table in which it lists all the surrogate endpoints they have ever considered or will consider in the future. And it mentions that a surrogate, if the surrogate endpoint is known to predict clinical benefit, then it can be used to support regular approval. If it is not known to predict clinical benefit, but is reasonably likely, then it can be used to support accelerated approval. So basically, if the evidence is strong that the correlation is strong, then it can be used to support regular approval. If the evidence is weak for that correlation, then it can still be used to support accelerated approval. There is no problem with this. This is a good approach. Now, if we look at the table, however, the table looks quite unusable, because this is what the table looks like. It lists all the disease here on the first column. It says, for example, cancer, solid tumors, and then it includes all types of solid tumors here, and then says response rate can be used for accelerated or traditional approval. Now, why I'm saying this is quite unusable table is because you would think that response rate has different correlation for breast cancer versus ovarian cancer versus renal cancer versus pancreatic cancer versus colorectal cancer. It cannot have the same level of surrogacy for each and every tumor type. And therefore, this response rate may be used as an accelerated approval for ovarian cancer, but may not be used as a traditional approval for hepatocellular cancer. I'm just giving random examples. But the bigger point I'm trying to make is we can't put in all solid tumors together and say that we will use response rate for accelerated or traditional approval. This is quite unusable. So, okay, I'm highlighting here breast cancer. So for breast cancer, we have the breast cancer is included here under 5 different rows. So response rate is here, progression-free survival is here, disease-free survival is here, event-free survival, and pathological complete response. So pathological complete response is unique in that only breast cancer is listed here and no other tumors. So the question is, as you remember from here, it says that the surrogate endpoints must be validated. So the question is, how do we validate a surrogate endpoint? So how does that validation process works? So how do we validate a surrogate endpoint? Of course, we take a number of trials. For example, I have 5 trials here, and then you plot surrogate endpoint on one axis, and then you plot the clinical endpoint on the other axis, and you see whether improving the surrogate endpoint also improves clinical endpoint or not. So you look at the correlation between the y-axis and the x-axis. But in real terms, in research terms, what we actually do is, okay, I'll give you an example. This is from a different paper. So what they looked here is, they looked at OS, overall survival in months in the y-axis, and progression-free survival in months in the x-axis, and they looked at the correlation coefficient. It was 0.64. So this is a graph of OS versus PFS, and each point represent each trial. So they took a number of like 20, 30 trials, and they looked at the correlation between OS versus PFS. But is this what a surrogacy validation looks like? No, it does not, because this does not take treatment effect into account. And this is a little tricky concept. So what this basically means is a patient that has, if PFS is longer, overall survival is longer. To say if PFS is longer, overall survival is longer, and to say if a drug improves PFS, it also improves OS, are 2 different things. Now, what I'm trying to say is, for a patient whose tumor has responded, that patient may live a longer time. Response may lead to a better survival. But a drug that increases response rate will lead to improvement in overall survival is a different thing. And that's what is the difference between a prognostic marker and a surrogate marker. For example, let's say in breast cancer, if the hormone receptor is positive, then a patient who has a positive hormone receptor will live longer than a patient who has a negative hormone receptor, irrespective of treatment. Even if the treatments were the same or both the patients did not receive any treatment at all. It does not take treatment effect into account. It is just a prognostic marker. If you have that marker, you live longer. If you don't have that marker, you live shorter. But that's not what a surrogate validation is. A surrogate validation should test if using the drug to increase that marker will lead to improvement in survival or not. So the correct way to test is to test treatment effect rather than just the surrogate and overall survival. So how do we do that? The first thing we can do is we can test the difference instead of the median PFS. We can test the difference in PFS between the drug arm and the control arm and plot it on the x-axis, and the difference in overall survival between the drug and the control and plot it on y-axis. So this is a better marker. Or we can look at the hazard ratio, because hazard ratio gives us treatment effect. So we plot hazard ratio for surrogate on one axis and hazard ratio for survival on other axis. And then we look at the correlation. So R is called correlation coefficient, and R square is called coefficient of determination. But basically the idea is if this correlation is more than 0.85, then we consider it as a strong correlation and the surrogate is considered to be a valid surrogate for overall survival. Or if the R square, it's just the square of this, so it comes out as 0.7. If the R square is more than 0.7, then it is considered a strong surrogate. Now, these cutoff values for 0.85 or 0.7, they come from a German group. What is the cutoff that the FDA uses? Nobody knows. We have not been told. It is not. It has never been told what the cutoff the FDA uses. So that is one of our points in our paper that we discussed. The FDA should let us know what is the cutoff that it uses to consider a surrogate as a good surrogate. Maybe it uses a different cutoff for considering a surrogate a good surrogate for regular approval versus a surrogate as a good enough for accelerated approval. Maybe it's 0.85 for traditional approval and 0.7 for accelerated approval, but we don't know. And that is something that we should know. So a couple of important concepts is that, as I mentioned here, a good patient level surrogate, improving PFS, a longer PFS leading to longer OS, and a good trial level surrogate, a drug that improves PFS also improves OS, are two different things. So there can be a good prognostic markers, but in order to be considered as a good trial level surrogate for using the surrogate as a marker in clinical trials, it has to be predictive of treatment benefit, as we showed in previous graphs. So, sorry, I forgot to give a link to the paper that we published recently on this. I can send it later. So these all tables come from that paper. So we looked at all the surrogate endpoints that were listed for breast cancer, like in this table. As I showed, breast cancer was listed for response rate, PFS, DFS, EFS, and pathological complete response. So we wanted to check what the evidence is for these surrogates for breast cancer. And this is what we found. For pathological complete response, we found that there were two studies that conducted such validation studies, such surrogacy validation studies. And you may remember that we mentioned R square should be more than 0.7 for considering a strong correlation. But these were the R squares that the studies have found. One study found an R square of 0.09, and the other study found an R square of 0.24. So these are quite low R squares compared to 0.7, that is the benchmark. This is not even 0.1, and this is only 0.2. These are very, very small level of surrogacy. So this cannot be considered. So pathological complete response cannot be considered as a surrogate for overall survival. Again, a patient who has pathological complete response will have a longer OS because it is a prognostic marker. But a drug that improves pathological complete response rate does not lead to improvement in overall survival. So it is not a good surrogate marker. So this cannot be considered for approval. This is quite poor level of surrogacy. What about disease-free survival? For disease-free survival, we saw that there were again 2 studies. One study had an R-square of 0.38, but a different study had an R-square of 0.75. Now this meets the benchmark, right? This is more than 0.7. And this was for the adjuvant trials of HER2 positive early breast cancer. So for HER2 positive early breast cancer, DFS is indeed a good surrogate for OS and can be considered for approval. But except for HER2 positive early breast cancer, if you take all other types of cancer, then the surrogacy is poor. And in those cases, DFS cannot be considered as a surrogate endpoint for regulatory approval. What about response rate? So for response rate, again, there were 2 different studies conducted. One study showed an R of 0.5, and the other study showed an R-square of 0.3. Again, these are very low levels of correlation, so response rate does not correlate with overall survival. For progression-free survival, there were 7 different studies, a number of studies. And if you look at what they report, R or R-square, you see that it's 0.48, 0.49, 0.3, 0.5, 0.6. Only one study has an R of 0.7. So all of them show just moderate, mild to moderate correlation. None of them showed strong correlation. So the conclusion was that PFS was not strongly correlated with overall survival. And then finally, we have event-free survival. And actually, it is quite interesting that no study has assessed the validation of event-free survival as a surrogate endpoint. We could not find any study that tested EFS or the strength of correlation of EFS with OS. So the point is, this FDA table puts breast cancer in these 5 different places. And the FDA's own mandate is that the surrogate endpoints must undergo a validation test. And if the validation is strong, we can use it for traditional approval. If the validation is weak, we may still use it for accelerated approval. There is no problem with that. But the problem is, as we just saw, it lists breast cancer here, and here is response rate, and says it can be used for accelerated or traditional approval. We just saw that response rate has very weak correlation. So how can this be justified? Same is true for PFS. For DFS, again, for one particular subgroup, HER2 positive breast cancer, it was a strong correlation. So it could be used for approval. But for other subtypes, again, it cannot. And event-free survival, surprisingly, it has no validation test undergone until now, at least published validation studies. If the FDA has unpublished studies that they conducted, then that's a different story. But there is no published study testing the validation of EFS with OS. So how can this be included here if there is no such validation studies happening? And for pathological complete response, we saw that it has very poor correlation. So at least they don't mention traditional here. They mention it as accelerated only. So what we actually propose, and another point is that in the FDA table, there is one more column that says mechanism of action, and it lists as mechanism agnostic for all of these solid tumors and for all of these surrogate endpoints. But I think it is not mechanism agnostic, because we see that for different subgroups of cancers, the correlation is different. We just saw that for HER2 positive breast cancer, DFS is a strong predictor, is a strong surrogate. But for other types, it is not a strong surrogate. So I don't think we can just lump all of these tumors and surrogates together and say these are mechanism agnostic. So what I think is that we should the FDA table should actually be modified to include the surrogacy evidence for each tumor type, and that should be included in the table itself. Like when this table says breast cancer, like each solid tumor should have a different column. Instead of just saying solid tumors, here should be breast cancer, and here should be response rate, and then it should list the surrogacy standard for each surrogacy, for each surrogate marker, and should say that this has a this has an R-square of 0.8, so we are listing this as a regular approval. This has an R-square of only 0.4, so we are listing this as an accelerated approval. This has an R-square of only 0.2, so it is not included at all. That is what the table should say. That is what would make this table more usable and more important. And I think, again, that table should be, that evidence should be live, right? Because as you can see, these are studies, some of them are conducted in 2008, some in 2015, some in 2019. So as we get new trials, there are new PFS and OS. So there are new, those trials to plot on the graph. So with each new trial, these numbers, these R-scores may change. So a surrogate we thought was a good surrogate with more number of trials may turn out to be a poor surrogate. A surrogate that is a poor surrogate now with more number of trials may turn out to be a good surrogate. So this can't be such static studies conducted in 2015 and we just look at it in 2020. This should be updated and this should be live, and that should be included in the FDA table. So the questions after we completed this study, the questions that came to our mind was, first, is the FDA not fulfilling its own mandate? Because the FDA says the surrogates must undergo validation studies, but as we sort of event-free survival in breast cancer, some of them have not undergone those validation studies, and some of them have undergone the studies and showed that they are quite weak surrogates. The other issues is that with pathological complete response, especially, the correlation was so poor, less than 0.1, that it does not look suitable even for accelerated approval. And for the other surrogate endpoints, they look suitable for accelerated approval only. The correlation was not strong, not for traditional approval. Of course, the exception again is DFS in HER2 positive early breast cancer. So this was the conclusion. But what's happening is that we are so much focused on how many drugs we approve every year rather than whether those drugs actually improve clinical outcomes or not. So we published this paper last year, is the number of cancer drug approvals a surrogate for success. So the trend now, this is what we found, the trend now is that this pale blue line is the number of cancer drugs approved on the basis of surrogate endpoints. So you see that since 2016, in 2017, 2018, the number of drugs that are approved on the basis of surrogate has really spiked compared to 2016. It has really spiked. But if the surrogacy is weak, you'd expect that all of these approvals were accelerated approvals, and then they would be confirmed later in a clinical benefit. But what actually is happening is this maroon line is regular approval. So you'll see that even the maroon line is increasing. There is a spike. And accelerated approval, this green line, is actually decreasing. So what this actually means is more and more drugs that have shown benefit in surrogates only are now getting full approval rather than accelerated approval. And that's a problem, because we just saw that the surrogacy is very weak in almost all cases. And if it is weak, then we should at least give accelerated approval and ask for a confirmation of benefit. If we are giving regular approval upfront, then these drugs are never going to be tested in a confirmatory trial. So we'll never know if they might even harm, and there might be a detriment in overall survival. We'll never know these answers. So, you know, this type of, this is not how we should fix the issue of drug approval. This is a wrong way to fix it. Similar to what this cartoon shows. This is not how you fix a broken rib. But this is, I think, what's happening right now in the regulatory science. Most of us might remember what happened with bevacizumab in 2011 when FDA decided to revoke its approval because it got accelerated approval on the basis of PFS, and then OS was not confirmed in a confirmatory trial, so its approval was revoked. That is how an accelerated approval mechanism should function. But you'll see that this happened in 2011 December, but in 2012, there was a trial of everolimus published in breast cancer. And they published only PFS at that time, and there was a good benefit in PFS. The hazard ratio was 0.36, and bevacizumab also had similar story. It had good benefit in PFS. Now, you'd think that they would learn the lesson from bevacizumab, but here they did not. And based on this PFS benefit only, they gave full approval instead of accelerated approval to everolimus. Complete approval. And in 2014, just 2 years later, the overall survival data were published for the same drug. And now we see that the overall survival was not, there was no benefit in overall survival. So this is just like bevacizumab. But the difference is bevacizumab had accelerated approval, so it got revoked. This received full approval upfront, although the surrogacy was weak, and we just learned from bevacizumab. This got full approval upfront. So when the overall survival results were published, there was no question about revoking its approval because it was not an accelerated approval. And as recently as last year, alpelisib received approval based on improving progression-free survival in a subset of patients. Again, this was a full approval. This is not an accelerated approval. So in future, if we see that overall survival was not changed at all, the approval is going to stay. So my conclusion is that the FDA table of surrogate endpoints should be revised. As of now, it keeps everything together and does not give any evidence of surrogacy, so it's quite unusable. Each tumor should have a different row because the same surrogate may have different correlation in different tumor types. Each surrogate listed should have the evidence for why that surrogate is being listed on the table, and the table should be made live by updating that evidence as new information arrives. This is the conclusion for that paper, but I think there is a better solution as well, a solution in which we don't need the concept of surrogate endpoints at all. The surrogate endpoints is creating a lot of confusion. So the simple solution to end this debate is, the reason we are using surrogate endpoints is for time, right? That's the only thing. Otherwise, we all want to know whether the drug improves survival or quality of life. The only reason we are using surrogate is to save that 11 months time. So I think the best way moving forward is to test every new drug in the last line of therapy as fourth line or fifth line, the last line of treatment, where the comparator is placebo, nothing, because there is no treatment. All treatment options are exhausted. So in the last line, the life expectancy is sought, the survival is sought, and the comparator is placebo. So if we test a new drug in the last line of therapy, then it does not take a long time to see an effect on overall survival. We can see that the drug improves overall survival or not in the last line of therapy in a relatively quite short time. And if it did improve overall survival, then you can now bring that drug in previous lines, in first line, second line, and you could just improve PFS. And that would be fine. But the drug should have evidence of improving survival, at least at some point, at least in some line of therapy. So we could start from last line. It would take quite a short time to improve survival against placebo in the last line of therapy when everything is exhausted and survival is sought. And then we could bring it up front and test for other endpoints that would fulfill both the mandates. And why I'm saying this is because we looked at this bevacizumab saga, and we saw that we found actually 8 clinical trials for bevacizumab in breast cancer. And we looked at whether PFS correlated with OS for this particular drug in breast cancer, for bevacizumab only, instead of looking at all the drugs. And after 8 clinical trials, our answer was, we don't know, could be. So there is uncertainty in the surrogacy for a drug even after we have 8 clinical trials. Nowadays, we don't have even 8 clinical trials. We have just 1 trial. The drug is never tested again. So this was a unique case in which we had 8 clinical trials for the same drug. And if we are not certain about the surrogacy of a drug, even after 8 clinical trials, then the shortest answer would be to just do one trial, but test for overall survival, and then you know whether it improved overall survival or not. Otherwise, you would do 8 trials and still not know whether it improved overall survival or not, because you never tested for that. So moving forward, I think we should test the drug for improving overall survival, at least in the last line of therapy. It should improve overall survival. If it cannot improve overall survival versus placebo as a last line of treatment, then it is probably a bad drug, not worth the side effects and toxicities and cost. And then when bringing the same drug to earlier lines, in that case, maybe we can just test for progression-free survival. And the other important thing that we need to advocate for is we need to ask the FDA what its level of evidence requirement is. For what level of surrogacy it thinks is regular approval okay, and for what level of surrogacy the FDA thinks it can give only accelerated approval, and that evidence should be made clear. Just as an end note, towards the end of the presentation, I think I want to say a couple of things about COVID-19 because that's the pandemic we are living in right now. I think there are a couple of lessons for us to learn from COVID-19. COVID-19 is a pandemic, but in a sense, I think cancer has always been a perpetual pandemic, although it's not infectious. If people don't put the numbers of people dying from cancer on TV every day, and that's the reason why we are so complacent. If we put the numbers of people dying from cancer every day on the TV, then we would think we'd really be scared. And we'd do something about it, like we're doing for COVID-19. And the lesson for surrogacy is that so many drugs are being tested for COVID-19, and for right reasons, we are excited only about dexamethasone because it improved overall mortality. It saved people from dying. There are a number of other drugs. Some are testing whether it improves oxygen saturation by 5% or not, and nobody is excited about those drugs. So why do cancer patients have to settle with a lower level of evidence then? Shouldn't patients with cancer, don't they deserve the same level of evidence that, see, this is a drug that will definitely improve your life or your quality of life? I think that's the benchmark that we should all aspire to. Thank you. Now I'm happy to answer your questions if there are any.
Thank you so much, Dr. Gawali. We will now start the Q&A and discussion portion of our webinar. As a reminder to the participants, if you would like to ask a question, please click on your hand icon on the bottom of your screen and we'll call on you. You may also submit questions via the Q&A feature, and we welcome your questions. So let's see, we have about 8 questions in the Q&A, and I guess what I'll do is I'll just start going through them. I'm going to take the first question from Helen Schiff. If you give a drug accelerated approval, won't people drop out of the confirmatory to cross over to the drug?
Yeah, the issue of crossover is important in trials. With accelerated approval, as I mentioned, what usually happens is the accelerated approval, for example, is in second-line setting, but the confirmatory trial is conducted in first-line setting. So it's conducted in a little different line so that once it gets approved, it also receives expanded indication besides confirmation of benefit for the accelerated approval. And the issue of crossover is important. But, you know, once a drug has been proven effective in, say, second line, and we are testing the drug in first line, then there should be crossover because the patient will need access to the drug after disease progression. So in that case, crossover is important. But in case where the drug has never been proven, then the idea of crossover is not to deny an effective treatment to patients with cancer. But so A, when the treatment is proven to be effective, then there should be crossover. But B, when the treatment is not yet proven to be effective, then in that case, at least in any line of treatment, then in that case, crossover will actually hamper the results of the trial. But with regards to if the drug is already available under accelerated approval, won't all patients just get it, and why would they enroll into trial? That is another important question. And that's the reason why uh, the confirmatory trial should begin before the drug receives accelerated approval. And actually, FDA has had published a paper in 2018 in JAMA Oncology about their own experience with accelerated approval. And even that paper confirms that for those drugs whose confirmatory trial had started before getting accelerated approval, those confirmatory trials confirmed benefit quite early, like years earlier than other trials whose confirmatory trial started only after getting accelerated approval. So that question is quite right, that if patient already has access with accelerated approval, they might not want to participate in trial. But the solution is that the confirmatory trial should have already started by the time the drug got accelerated approval.
Great, thank you. Jane Perlmutter, did you want to ask one of your questions?
Sure. I've been involved. Can you hear me?
Yeah.
I've been involved with some researchers who are looking at endpoints, and they want a slight modification of overall survival. So instead of using overall survival, they want to exclude any deaths that are caused by a second primary, not the initial one, or causes that they don't think are associated with the drug. And on the one hand, this will tend not to dilute any effects of efficacy, but on the other hand, it might fail to find some hidden toxicity. I'm wondering your reactions to that.
Yeah, that's a very important question. Thank you for asking. The issue of cancer-specific survival is a little tricky, and there is a reason why we are not using it, because the beauty of overall survival is in that it measures both efficacy plus harms at the same time. So if there was a cancer drug that had very good efficacy in controlling the cancer, but for some unknown reason, let's say it leads to increased suicide rates among patients, then such type of harm would never have been captured if we did not measure overall survival, and if we measure only cancer-specific survival. So overall survival, the beauty of overall survival is that it captures the whole benefit plus harms within one metric. And I think cancer-specific survival will have a lot of pitfalls, and we should be careful about using it.
Liz Frank, you had a really good question about quality of life outcomes.
Yes, please.
Maybe I should just.
I think, am I unmuted now?
You're unmuted.
Oh, okay. So my question was that quality of life outcomes are oftentimes, even if they're included, they're underpowered. So, and I guess I'm thinking that's one of the not very good excuses for why they're not reported with the primary outcome. But I'm wondering what your thoughts are as advocates about when the quality of life outcomes should be reported, and what we can do about the fact that there's oftentimes a power issue with those outcomes.
Yeah, that's a really good question. And as a disclaimer, I have been involved with the ESMO Quality of Life Working Group, and we are trying to formulate, I don't know whether you are aware or not, there is something called the ESMO Magnitude of Clinical Benefit Scale to measure the value of cancer drugs, and it gives the rating of cancer drugs based on how good the clinical efficacy is. So similarly, we also have a Quality of Life Working Group. The results are not published yet, but we also want to standardize the reporting of quality of life outcomes. I totally agree. It is all haphazard right now. There are different tools to measure quality of life. There is obviously power issue as well, because quality of life has never been the primary endpoint. It is always a secondary endpoint, and people report it usually descriptively without looking at the significance. And then there are a number of tools and different tools. So how do you compare a drug that improves quality of life in one particular tool versus a different drug that improves quality of life in different tool? And then again, even within quality of life, there is the question about the global quality of life improvement versus improving quality of life only in particular domains. For example, improving quality of life in terms of pain control and anxiety, but degrading quality of life in some other parameters, for example, mobility. So this is quite a complex issue, and I know that the FDA has also been working to harmonize this, but I guess we are all waiting for, let's say, the first cancer drug to be approved on the basis of improving quality of life, so that might open more and more research and insights into this. Because as of now, for a cancer drug, if you think in terms of industry, then you don't have any motivation to measure quality of life right now. If you measure PFS, you'll get approval straight away, irrespective of quality of life. So there should be some mechanism by which there is an onus on the sponsor of the trial to also measure properly quality of life. For example, if we said a drug that improves PFS will receive approval only if it improves quality of life, then maybe there will be more um, encouragement for everyone to get involved.
Well, thank you. And maybe when that paper is published, maybe Michelle can send out a link to it. It sounds like it would be very interesting.
Yes, I'll definitely send it to Michelle.
Do we have time for one more question?
Yeah, I do.
Oh, okay. Tracy Solek, are you still on?
I am.
Did you have a question you wanted to ask?
Oh, sure. You were talking about testing a drug for the first time in the last line of therapy. But if we do that, then you're testing it in patients who are really, really heavily pretreated and may have may be immunocompromised, may have morbidities from their prior treatments. Is that going to really be reflective of how the drug might perform in an earlier line?
Yeah, so my proposal was that the drugs should be able to show improvement in survival at least somewhere. So the best, from the drug's point of view or the sponsor's point of view, the easiest would be to test it in the last line of therapy because the survival is shorter. But that would not automatically give approval for the drug to be used in first line. The drug has to be tested in a different trial in first line, but now when they are testing it in the first line, they could simply show an improvement in progression-free survival because of all the pitfalls of showing improvement in overall survival in the first line because of the possibility of multiple lines of treatment. So the easiest thing would be that you show that the drug does improve survival in the last line. So that means the drug is indeed efficacious, and then you show that it improves PFS in the first line. So that would cover both sides of the equation. But right now what's happening is we have so many drugs that have never shown improvement in overall survival at all, even in the last line. Like, we don't know whether it actually has any role or not.
Okay. Vernal, are you still on? Vernal Branch? I'm just going to read it for the sake of time. It would seem that these tumors should have genomic profiles that are similar in order to test these drugs.
Sorry, I didn't understand the question.
Yeah, I don't think maybe this is not a completed question. I'm going to move on to the next one. Amy Delson asks, what about PCR in the neoadjuvant setting, basis of the I-SPY trial with many adapting that surrogate endpoint?
That's a very nice question. Yeah. So PCR has been used in the I-SPY 2 trial. As far as I know, no drug has been approved on the basis of I-SPY 2 alone. They are moving on to phase 3. So I-SPY 2 is being used as a way to filter drugs and choose only those drugs that have a good probability of success and test them in a phase 3, because of course phase 3 is resource intensive and you don't want to test a drug that may fail in phase 3 unnecessarily. So I-SPY 2 is being used for that purpose, and I think if we are using PCR as a filter to find the drugs that have greater likelihood of success in a phase 3, I'm happy with that. But if we're using PCR as a means to lead to drug approvals, then I'm quite not happy, because as we all saw, the correlation is quite poor there.
So Michelle, there's other questions. Do you want us to keep going, or?
Yeah, I still have a couple of minutes if you all have time.
I think we've already taken up too much of your time, but what I may do is follow up with some of the questions and see if we can get some written answers that we can share with people. But
Yeah.
At this point, I just want to thank you, Dr. Gaiwali, for your time, your expertise in the presentation today. I also want to thank everybody who joined and asked questions. They were great questions. I'm sorry we didn't get to all of them. I would like to also remind people, as you exit the webinar, if you wouldn't mind completing the brief evaluation, we'd very much appreciate that. And I do also want to announce that we do have our next LeadCast webinar, which will be presented on July 29th. We'll have Dr. Jonathan Kimmelman, who's going to be talking about the ethics around randomized controlled trials and clinical equipoise. So you'll receive more information about that in the coming weeks, and we hope you'll be able to join us for that. But again, thank you so much, Vishal, for joining us and sharing your expertise. It's always a pleasure to have you, and hopefully we'll have you again in the future. No doubt I'll be asking, but
Yes,
so with,
it's been my pleasure to speak to the NBCC team.
right, well, you're always welcome, and hopefully we can maybe even have you at our summit next year. So with that, I want to thank everybody, and that concludes the webinar. So thank you, everyone.
Thank you. Bye.
Bye.
Bye.